All right, diving right in today.
Right into it.
We're tackling something pretty amazing, I think.
Yeah.
How a baby gets their very first microbes, in their gut.
Yeah, yeah.
Kind of like setting up, a garden that will be there for life.
Right.
And we're really zeroing in on how mom plays a super direct role in that,
Yes.
which scientists call vertical transmission.
Yeah, exactly. And, the map we're using to explore all this microscopic world, it's
Yeah.
this really in-depth study called, the title's a mouthful, Studying Vertical Microbiome Transmission from Mothers to Infants by Strain-Level Metagenomic Profiling.
Wow, that's quite a title.
It is quite a title, but basically it gets down to the nitty-gritty of which microbes are moving from mom to baby.
Okay, so for you, the learner, listening in.
Yeah.
Our mission today is to go way beyond just like naming what kinds of bacteria are hanging out, together.
Right.
We're going to get down to like a super detailed level,
Ew.
like the genetic variations, like the strains
Yes.
of these microbes. This study is hardcore science, I got to say.
Yeah.
Not just confirming these transfers are happening, but giving us clues about how they start to shape a baby's gut as it's developing.
Exactly. Yeah.
Okay, so first things first.
Yeah.
Why are we so obsessed with the infant gut microbiome in the first place?
Well, it's really the foundation already.
Okay.
The infant gut microbiome is working overtime from the very beginning.
Wow.
It's vital. It's crucial for so much. I mean, first off, think about helping baby digest their food,
Okay.
right? Getting all that good energy out of it,
Right.
and then setting up their metabolism.
Uh-huh.
But here's the thing, and especially early on, it's a teacher for the immune system,
Oh, wow.
right? Helping it learn what's a good guy, what's a bad guy.
Right, right.
And even keeping those bad bacteria in check
Okay.
just by taking up space and, using up resources.
Just it can't get a foothold, is that what you're saying?
Exactly, exactly.
I mean, it's crazy to think about all the work going on down there.
It really is.
Invisible, until you kind of really look.
Yeah.
And when you were talking about preventing infections, it reminded me of, what they're doing now with those microbiome transplants.
Right.
For those really tough C. diff infections.
Oh, yeah, yeah.
That's like major gut power.
Right, absolutely. I mean, that kind of therapy, it really shows you how influential the gut microbiome can be.
Right, right.
And so you can see why understanding how it gets going in infants is kind of a big deal,
Very
right?
important. Yeah.
And, could have an impact on health throughout life.
Yeah, that makes sense. What do we kind of generally think about how babies got their microbes from mom?
Earlier studies, they used methods like culturing bacteria in the lab
Okay.
and analyzing specific genetic markers, this thing called 16S rRNA profiling.
Go ahead.
Gave us, like a first look, a broader picture.
Okay.
And they pointed to mom passing on microbes to her infant. Breastfeeding seemed like a big way that was happening, direct delivery through the milk.
Right.
And then how a baby's born mattered too.
Okay.
Vaginal birth, baby picks up some of mom's vaginal microbes,
Makes sense.
which wouldn't happen with a cesarean.
Okay, yeah, that makes sense, just kind of contact-wise
Exactly. Yeah.
during birth and feeding.
Yeah.
But didn't you say those earlier methods had a limit?
Yeah, they did. They could identify the species of bacteria there,
Okay.
but they often couldn't tell you the difference between different strains of that same species.
Okay, I'm not sure I get that.
So think of it like knowing two people with the same last name,
Okay.
but you don't know if they're like close family or just, distantly related.
Oh, okay.
Yeah.
So
Yeah.
just because like you find the same type of bacteria,
Right.
let's say E. coli,
Mm-hmm.
in both a mother and her baby,
Yeah.
it doesn't automatically mean it came from her.
Right, right.
Like there could be lots of different versions of E. coli out there.
Exactly. Different people, they can have different strains of the same bacteria.
Okay.
So just finding E. coli in both doesn't mean there was a direct transfer.
Okay.
You got to show that it's the exact same genetic variant, the same strain.
Okay.
That's why this study's focus on strain-level analysis is so important.
Okay, so how did they do that?
Yeah.
How did they actually pinpoint these specific strains?
They had some serious tools. They had 5 mother-infant pairs,
Okay.
collected samples,
Okay.
fecal samples from both mom and baby,
Uh-huh.
and breast mix samples,
Okay.
all when the babies were about 3 months old.
Okay.
Then follow-up samples, 2 pairs at 10 months,
Okay.
and 1 pair at 16 months.
So kind of getting snapshots across that first, year or so of life.
Right, yeah.
Okay, so then what? What do they do with all that, stuff?
This is where it gets really high tech.
Okay.
It's called shotgun metagenomic sequencing.
Okay.
So instead of just looking at one gene or a few, this method, they look at all the DNA of all the microbes. Wow.
So you're getting like the entire genetic blueprint of the whole community.
That must be a ton of data.
It is. That's where computational power comes in.
Okay.
They use special software, PanPhlAn and StrainPhlAn.
Okay.
These are like microbe DNA detectives.
Wow.
They sift through it all and find even tiny genetic variations, single nucleotide variations. They call them SNVs.
Okay.
And they also see differences in the entire set of genes.
Okay.
So they can build like a unique fingerprint for each strain.
Oh, wow. That's pretty cool.
It is, yeah.
So microbial fingerprinting.
Yeah, exactly.
So then they take these fingerprints and they can compare them, right?
Exactly.
Between mom and baby to
Yeah.
see if there's a match.
Right.
Like CSI for microbes.
Yeah, pretty much.
All right. I love
Great.
that. And then they went even deeper, right?
Yeah. So for 2 of the pairs, at the later times, they used metatranscriptomics
Okay.
on the fecal samples.
Okay.
It doesn't just look at the DNA, the potential.
But at the RNA, which shows you which genes are actually turned on,
Right.
it's like seeing what proteins are being made actively.
Okay.
It's like you have the blueprint of a factory,
Right.
but now you see which machines are running.
Oh, I see.
Yeah.
So, okay, they did all that.
Yeah.
What did they actually find?
Well, the results were really interesting.
Okay.
They found specific strains of bacteria, like bifidum,
Okay.
Coprococcus comes, and Ruminococcus bromii were some of the standouts
Okay.
that were in samples from the same mother and infant pair,
Okay.
but were genetically different from strains in other pairs.
Okay, so like a baby would have, let's say, B. bifidum.
Yeah.
That was basically identical to its mother's,
Yeah.
but totally different from the B. bifidum in other babies in the study.
Exactly right.
Wow.
In one pair, their B. bifidum strains were 99.96% identical.
That's almost a perfect match.
It's incredibly close, yeah.
Yeah.
And when they compared these similarities within a pair to the differences between pairs statistically,
Okay.
it was very strong evidence of a link.
Wow.
It wasn't just random chance.
Yeah, okay.
Yeah.
And then they looked at the whole set of genes too, right?
They did, yeah. They used something called pangenome analysis,
Okay.
and they confirmed that the shared strains between mom and baby
Okay.
also had the same unique collection of genes.
Okay.
Even more support for the idea of a direct transfer.
Wow.
Now, they said you can never totally rule out, maybe they got it from the environment.
Right.
But these genetic markers all pointing the same way, it makes vertical transmission pretty likely.
Okay.
Yeah, on average, they saw this kind of strain sharing for about 14% of the species.
Okay, so 14%.
Yeah.
That might not sound like a lot,
Yeah.
but when you think about how many microbes are in there,
Right, exactly.
14% could still be a lot of microbes.
It's a significant contribution.
Yeah, with maybe big effects down the road.
Exactly. Even a small percentage at the strain level can mean a lot of microbes setting up shop.
Okay, what about breast milk?
Yeah.
Did that turn out to be a major route for this, like, strain transfer?
The milk samples were tricky.
Yeah.
There were molecules that got in the way, made it hard to read the DNA,
Right.
but they got most of it analyzed.
Okay.
And what they found was milk had less diversity of microbes than the fecal samples at 3 months.
Okay.
They did see some bacteria that's usually on skin, like
Huh.
Corynebacterium krupenstedti and Staphylococcus epidermidis.
Which makes sense, right?
Yeah, given where it is.
Close to the skin.
Exactly.
Yeah.
But these weren't in the baby's guts so much.
Okay.
So maybe not the main colonizers there.
Okay.
Interestingly, at the later time points, they saw more Bifidobacterium biface
Okay.
and some oral bacteria,
Okay.
Streptococcus and Phalanella in the milk.
Oral bacteria.
In the milk. Yeah. The thinking is this could be from what they call retrograde flow
Okay.
during breastfeeding. So some of the baby's saliva and its microbes
Mhm.
might go back up into the gland.
Oh.
But they didn't have any oral samples from the babies in this study.
Okay.
So couldn't really confirm it for sure.
Right.
So milk definitely has bacteria in it.
Yeah.
But this research suggests that maybe the key colonizers of the gut are coming from other places.
Okay.
Or maybe those milk bacteria establish first, but then get replaced by strains that are better suited for the gut.
Okay.
Okay.
So what happens to those vertically transmitted strains?
Yeah.
Long term, I mean,
Yeah.
are they like there to stay?
Well, they saw some interesting things with the multiple time points.
Okay.
Some strains were shared early on,
Right.
but then got replaced later in the baby's gut.
Okay.
For example, in the 16-month-old,
Okay.
the R-bromi strain at that point was different from the one at 10 months,
Okay.
and neither matched mom's strain.
Wow.
They saw similar stuff with B. brevi and B. longum.
Oh, okay.
Now, this is based on a small number, of course, but it does suggest that getting a strain from mom doesn't guarantee it'll be there forever.
Right.
It's a dynamic environment, that infant gut.
So it's not just, one transfer and then that's it.
No.
It's like an ongoing process.
It is, yeah.
Okay, now the study looked at viruses too, right?
They did, yeah. The virome, the viruses, that's important too.
Okay.
And bacteriophages, those are viruses that infect bacteria.
Right.
Those were the main DNA viruses.
Okay.
Lots of phages from the cautovirales order, which are common in the gut.
Okay.
They also saw a link between crassphage, a really common phage, and bacteroids bacteria, which supports what they think about kind of which bacteria it likes to infect.
Wow, so bacteria even have their own viruses.
They do. It's an ecosystem within an ecosystem.
That's amazing. What about RNA viruses?
Oh, that was really interesting.
Okay.
They found this pepper mild model virus, PMOV, an RNA virus.
Okay.
At really high levels in 2 of the pairs.
Okay.
And what was really cool, the exact same strain of PMOV was in both the mother and the infant.
Wow.
In those 2 pairs.
Okay.
And different from other known PMOV strains.
So pretty clear vertical transmission, even for a virus.
Seems like it, but got to be careful here.
Okay.
PMOV is a plant virus.
Okay.
It's common in like chili peppers and stuff.
Okay.
So since these babies were probably eating some of that,
right.
You could say they both got it from food.
Okay.
So it's interesting, but they can't say for sure it was vertical transmission like they could with some bacteria.
Right. You got to think about all the ways something could get in there.
Yeah.
Especially with diet.
Exactly. Yeah.
So they track the microbes, they track some viruses.
Yeah.
What about what these microbes are doing?
Ah, good question.
Right. Are they just hanging out?
Right.
Or are they like
right.
working?
Yeah, they analyzed the predicted functions of the communities, based on the genes they had.
Okay.
Lots of overlap between mothers and infants.
Makes sense, right?
It does, yeah, given the shared microbes.
Right.
But there were also differences,
right.
Probably because the adult and infant gut are different environments.
Right.
For example, the infants, they had a higher potential for breaking down mucin.
Okay.
That's part of the gut lining.
Okay.
And for making folate, an important B vitamin.
Okay.
But they weren't as good at breaking down complex starches.
Okay.
Which makes sense, since they're mostly drinking milk.
So it seems like the baby's microbes are geared towards what they're eating.
Exactly. And the older infant who had started weaning,
okay,
their microbiome was starting to look more like the mother's.
Interesting.
Yeah, so diet and maturity of the gut play a role.
Okay.
Now, they also looked at actual gene expression and what the microbes were actively doing.
Huh?
And they found fermentation was much higher in mothers.
Okay.
Which makes sense. The adult gut is more anaerobic.
Right.
And starch breakdown was low in infants.
Okay.
Matching that lower potential they saw.
Okay, so what about those specific strains from mom?
Right, good question.
Were they actually doing anything in the baby's gut?
So they used this strain-specific metatranscriptomics.
Right.
And they found those strains like B. vulgaris, E. coli, B. bifidum.
Okay.
They were all active in both mom and baby.
Wow.
So they weren't just passing through, they were functioning members of the community.
Okay.
It means successful colonization.
That's pretty amazing because the transfer isn't just happening. It's like sticking around.
Exactly, yeah.
And they saw differences in the activity of the same B. bifidum strain between the younger and older infants.
Right.
The older one, less activity of that, usually infant-associated species,
right,
and more activity of R-bromii, which is more common in adults.
Yeah, so again, diet and age making a difference.
So taking it all in, what does this study mean for us?
It shows how powerful these technologies are,
Yeah.
Sequencing and analysis.
Yeah.
To really understand vertical transmission. We're not just seeing shared species anymore.
Right.
We're tracking specific strains from mom to baby.
Wow.
And even starting to see what they're doing.
That's amazing.
It sets the stage for bigger studies to really nail down how it happens.
Right.
And what the long-term effects are.
So for you, the learner, here's the takeaway.
Yeah.
Moms are giving very specific strains to their babies.
Yes.
That are active in their guts.
Right.
It's not just random microbes. It's like passing on blueprints.
Yes.
But it's not a one-time thing.
No.
And those first microbes might not always stay there.
Yeah, that's important to remember.
Being able to see this
Yeah.
at the strain level
Yeah.
is like a whole new way of looking at it.
It is, absolutely.
It really is.
Yeah.
And thinking about the future.
Yeah.
This kind of analysis.
Yeah.
Imagine doing it with way more families.
I know, right?
Looking at different parts of the body.
Yeah.
You know, not just gut and milk.
Yeah.
And even the role of other family members.
Exactly.
And the environment.
Yep.
It could really show us what shapes the infant microbiome.
It would be fascinating.
In those crucial first months.
Absolutely.
It makes you think about how those early exchanges.
Right.
This kind of inheritance of microbes.
Yeah.
Could have huge effects.
Yeah.
On our health.
Yeah.
And how we develop, you know, in ways we're just starting to understand.
And it goes so far beyond just the genes we inherit.
I know, right?
Incredible.
It really makes you think about, like, breastfeeding.
Yeah.
And the environment we raise kids in.
Absolutely.
What it all means for these microbes.
Yeah.
It's a lot to think about.
It is.
Thanks for taking this deep dive with us today.
Of course. My pleasure.
Really interesting stuff.
Yeah. Fascinating.
We'll see you next time.
See you then.
Bye.